From University of Maryland University College, this is Thursday Thoughts with Dr. Alan Drimmer. This series features faculty and friends of UMUC highlighting areas of academic expertise.
Welcome to Thursday Thoughts at University of Maryland University College. I'm Alan Drimmer, Chief Academic Officer here at UMUC, and today I'm joined by my friend, Professor Jim Coker, Program Chair in Bioinformatics, as well as Biotechnology Regulatory Affairs, and also the Director of our Ethics program on Bioinformatics. Today we're going to be talking about biotechnology, bioinformatics, and other related things. And I feel like I know something about this subject because when I was a kid, my favorite show was The $6 Million Man, starring Colonel Steve Austin.
Yes.
They rebuilt him, they made him better.
Yeah, stronger, faster.
Things like that.
Absolutely.
So I do really like the subject.
Okay.
That being said, I'm rather thin on current knowledge about it, so we'll be talking about that. So to start, could you tell us just how you got interested in this field and how you got to UMUC? I mean, it's a fascinating area, but what's your story? How did you come to embrace this?
So I guess the long story, maybe the long short story. So I started off like a lot of people that were interested in biology. I really wanted to be a medical doctor, right? So usually kids, when they start off, like, oh, I want to be a doctor. That's the first thing most people think about when they think about biology. And I realized after some time that, I really wasn't doing the things that I needed to do in order to get into medical school, like going to see the pre-medical advisor and all that kind of stuff. And about that time coincided with me starting to work in a lab. And I found out, I really like this. This is really fun. Like, you get to explore the boundaries of science and limits of knowledge and what we know and what we don't know about things. And as, 19 years old then, you can go into the lab and you can do experiments and you can really sort of figure out at a really young age sort of what we know and what we don't know and be a part of that process of figuring out, what's going on with life. So that did some research as an undergraduate that led to a PhD at Penn State, which led to a postdoc at the University of Maryland, Baltimore, at the Biotechnology Center, which is no longer in existence, but it was downtown Baltimore, which led to a faculty job in Alabama, which eventually led me to come back to Maryland to the current job. So that's the short, long story.
Wonderful,
yeah.
Wonderful. So you're teaching in the graduate school, you're
Right.
teaching these sorts of programs.
Right.
What sort of careers do your students typically go into or are coming from? Many people probably work in the field and are looking
Right.
for additional credential, but
Sure.
what are the kinds of jobs and things that people do related to this subject matter?
So let's talk about bioinformatics first, because there's that in regulatory affairs. So for bioinformatics, a lot of our students are already working in the field. They'll either be working in a lab somewhere or either in the private sector or in government. And they're basically coming to us to get a master's degree in order to further their career, to get ahead in the current job or the current area that they're in. If we're talking about regulatory affairs, we're one of a small handful, about let's say 20 or so programs that actually have a master's level degree in regulatory affairs. So a lot of our students are not necessarily in the field of regulatory affairs yet, but are trying to get into it because we're one of the few places that can give them a credential in order to get there. And a lot of our students those students want to go to work at some place like FDA or a pharmaceutical company or a biotech company to sort of help them develop medical products. But if they have a job currently, which many of them do, they're probably also working in a lab, working at a pharmaceutical company, probably actually doing bench science, but then are thinking, this regulatory stuff sounds great, so let me look at that too. And so we're there to help them with a credential to help them improve their career.
Right. One of the things, having not looked at this terrain for a while, I'm interested in knowing what the sub areas are, what the subtopics are under, let's just say if you think about biotechnology as a whole. Certainly, I've learned a little bit about bioinformatics. That seems like it would be one piece.
Sure.
What are some of the big sub areas that people think about with respect to biotechnology?
Right. So let's start with a broad definition, I guess, of what biotechnology is for people that don't necessarily know. And that's really basically just exploiting biological processes for an industrial purpose. And I know that's really broad, but biotechnology is really broad too. So that's about the best you can get for defining it. And since it's so broad, there's a lot of ways to break it down. You could break it down into subdisciplines. So you can say you've got bioinformatics, you've got molecular biology, you've got systems biology, you've got a lot of different types of things. My favorite way to break down biotech is with a color system. And this is a pretty well-known way of breaking down biotech. And so you basically break it down on all these colors. So you have red biotech, which deals with medical products. You have blue biotech, which deals with, let's say, like things like algae, which you will grow up to try to harvest their lipids in order to use them as alternative sources of fuel. You have green biotech, which would be looking at plants. And so when people think of biotech and plants, they usually think of GMOs, but it's not just GMOs. If you want to sort of look at what farmers have usually done with taking crops and crossing them with one another to get better yields, to get better resistance, that's also green biotech. You have white biotech, which is using enzymes for industrial purposes, which is pretty close to what I got my PhD in, so it's one of my favorite biotechs. And then you also have my absolute favorite biotech, which is yellow biotech, which is all the microbial products that make food. So if you think of wine, beer, and cheese, those wouldn't exist without microbes, they wouldn't exist without biotech, so that's yellow. So there's more, but we could go on with that for forever. So
what about bioinformatics? Where does that fit in? I guess that cuts across. Does that cut across all of those?
It does.
What color is that one?
So it doesn't have a color,
Okay.
but it does cut across everything. My cocktail party way of describing bioinformatics is if you think about if biology and computer science got married and had a baby, that baby would be bioinformatics. So it's basically taking computer applications and putting them towards biological problems.
Right. No, that's fascinating. I really didn't know much about that.
Yeah.
One of the things that I've noticed with respect to data analytics in general is that a lot of the work in data analytics is spent cleaning up the data. And
Sure.
Something like 80% of the work, in my experience, is spent on cleaning up the data, and then 20% on analyzing it. Is it the same in bioinformatics?
I'd say that's probably fair. I might modify that a little bit and say 60-40 or 70-30, depending on what it is you're actually doing. One of the reasons why bioinformatics became a field is because in modern biology, let's say a lot of people, when they think about biology, probably have this image of a person at a bench doing some experiments, maybe mixing some chemicals back and forth with one another. Modern biology, when you do an experiment, especially the type of experiments that I did when I was in my postdoc, you're literally generating millions of pieces of data with one experiment. And so for what I was doing as a postdoctoral researcher, you do that many times. So you may have 10 million pieces of data, and there's no way, no matter how smart you are and how good you are, that the human brain can process 10 million pieces of data. So what you need is some sort of computer-assisted way of doing that, and so that's really sort of where bioinformatics came out. Is things like looking at the genome and being able to process all of that information, being able to take gene expression data and process all of that. So all of that you get, to get back to your question about the percentage of how much time you spend grooming the data and how much time you spend actually analyzing it. That's where all of that comes from. So 10 million points, you're going to have to do a lot of grooming before you can do sort of the analysis work of that. Yeah,
sure. It's really interesting. It was counterintuitive to me. I just thought, well, people analyze the data, but actually it requires a lot of preparation and care to ensure that the data is sound.
Yeah, exactly. It's really important that when you do an analysis that all of your data is statistically significant, that it's supported by p-values, that it's real data and it's not erroneous. So whatever conclusions you draw are actually real, believable conclusions and not erroneous. Yeah, for sure.
One of the other things I've seen with data analytics is that some of the most interesting trends in data analytics involve the intersection of data analytics with some of the professions. So data analytics applied to finance, for example, or data analytics applied to healthcare, which I think is bioinformatics or related to it.
Or precision medicine, if you're talking about healthcare. Yeah, absolutely.
Or even, close to my heart, data analytics and education.
Sure.
And so on. Is that a similar theme or a similar potential growth area in your field? In other words, the intersection of bioinformatics or biotechnology with other areas? Does that, I don't know. That seemed to me like something very fascinating about.
Yeah, so we could talk for a long time about that. It's hard to answer your question only because I have to think of a small number of things since we only have so much time. And because of that, I think that I'm going to use a selfish example. And so here's my selfish example of this. So when I was doing my PhD research, What I was working on were enzymes that were from Antarctica, from organisms that were isolated from Antarctica. They are active in the cold, and we were trying to make these enzymes even more active in the cold. And so at this moment, you're probably like, well, who cares about that? Why does this relate to anything at all? So the reason why this relates is because the lab I was working in was also working with Clorox. And what they wanted to do were find enzymes that worked really well in the cold so that they could put them into detergents so anybody could use cold water to wash their clothes. And so if anybody out there has ever washed their clothes in cold water and your clothes came out clean, you can thank Clorox and researchers like me, selfish, I know, who studied all of these things to be able to find enzymes to be active in cold water, because most things are active either at room temperature or a little bit above. And so if you think about, let's say, stains, it's easier to get them out if the water's a little bit warm, but you can save a lot of money by washing your water or your clothes in cold water. And for that, you need enzymes to break down those stains. So that's what people like me do, is look at these enzymes, try to make them as active as possible at low temperatures. So everybody, when you wash your clothes, you can wash them at whatever temperature and get them clean, right? So another good sort of intersection of stuff is what I sort of mentioned earlier, which is precision medicine, which used to be called personalized medicine, and the people at NIH decided, well, all medicine is personalized, so we got to come up with a new name, which is precision medicine, which is basically looking at your specific DNA sequence and trying to develop drugs and biologics and medical products in order to target your specific disease. So let's say you have a particular type of cancer. When people talk about cancer, the lay public usually talks about, oh, well, I have brain cancer, I have pancreatic cancer. They talk about an organ. But scientists usually talk about the mutations that are the root cause of the cancer, because that could go anywhere. And it doesn't matter so much where it is, but it matters what the mutations are, because the medicines we're now developing really are targeted at specific mutations. And so that's basically what precision medicine is. And so then to sort of tie that back to bioinformatics, how do you know what mutations are associated with what particular type of disease? Well, that's bioinformaticians, and that's what they do. So they basically scan through all types of genomic data, and they look for small mutations that occur within the billions of letters that is the genome, and they say, okay, we see that this person has this particular disease, and also they have this particular mutation. And so they're probably correlated with one another. And they do that over and over and over again till you finally get a statistically significant correlation to say, okay, this particular mutation or this small set actually is associated with this disease. So now a pharmaceutical company, a biotech company, can come along and generate something that works or interacts with those mutations and then helps to cure that disease. Yeah.
Interesting.
Mm-hmm.
I'd like to touch on a number of sort of hot topics. There's so many interesting areas in your field that many of us know about or could be affected by. One is genetic research. That's some people hear about that. It's a big business, and certainly people have heard about pharmaceutical companies patenting drugs. But I wondered if can businesses patent genes or gene sequences or things like that? Is that a thing?
So no, that's the short answer. We probably want the longer answer too, but you cannot patent a gene. I believe it was in 2012, if memory serves correct, there was a landmark ruling by the Supreme Court. So this whole idea about whether you can patent a gene or not actually went all the way up to the Supreme Court of the United States to ultimately decide what you can do and what you can't do. The case, for a little bit of background about the case, the company that got these patents was called Myriad Genetics. Myriad Genetics spent a lot of time and a lot of money sequencing the BRCA1 and BRCA2 genes. And many people probably know that those two genes are closely associated with both breast and ovarian cancer. So the company spent a lot of money sequencing those two particular genes, and that led to them developing tests to determine whether or not women had these particular sets of mutations, which would possibly lead to these two awful types of cancers. So with all this work and all the money that Myriad Genetics had put into all of this work, they decided that they ended up getting a bunch of patents based off of all of that work. And so the United Pathologies Association decided that they would take Myriad Genetics to court over this, because basically all these patents meant is that Myriad Genetics owned the BRCA1 and BRCA2 genes. And everything that was associated with that. So it sort of would set up this type of monopoly where no other company could work on those 2 particular genes, but it would also mean that nobody could do any research on those as well without paying money to Myriad Genetics. So a lot of people got upset by this. This went through the courts for a long time. The lower level courts sided with Myriad Genetics. It eventually went to the Supreme Court, and the Supreme Court decided that they could not patent a gene because a gene was a product of nature. So you can't patent a gene, but you can patent a byproduct of the gene. So if you copy the gene, which is something called cDNA, you can patent cDNA, but you can't copy the DNA, the original DNA or the gene. That's the long answer.
Mm-hmm. And in genetics, are there other kinds of things that you can patent? Why, I don't know. I mean, that's an interesting court case that you mentioned.
Yeah.
Have there been other things like that relate to kind of proprietary ownership?
I mean, you can patent just about anything else. I think you can patent processes, you can patent enzymes, as long as they're... So you can patent enzymes that are modified in some ways. You can't, because if it's a product of nature, you can't patent an enzyme that you isolate from nature. But if you make some sort of change to it, like my PhD work, where you took a particular enzyme that was active at some level, you made some modifications to it, and now it's more active, so you patent the altered version of the enzyme and not the original version.
I see.
So the limit is basically, if it's a product of nature, you can't patent it, but pretty much everything else is fair game, especially if you're talking about the processes.
Right. Another thing you hear a lot about now related to genetics are these companies like 23andMe that
Sure.
Will analyze your genetic data and share it with you. People I haven't done it myself. I've had friends of mine who come to me so tickled that they find out that they're 1% this or that,
Yeah.
digging into their family trees. And it's
Sure.
very interesting, but it does prompt the question about what happened. What about that data? Is that data, certainly the individuals get it, what happens to that data? Do the companies own it? Is there anything, is that a concern? What happens to all this genetic data that people are serving up?
Right. So is it a concern? I guess that depends on the person that you're talking to. But no one should mistake the fact that 23andMe owns your data. That's clear when you get your kit. You sign away your rights to that particular set of data. We should also be clear about what 23andMe's business plan is. Their business plan all along has been to collect the data to sell it. So in that sense, they're not terribly dissimilar from Facebook. They're basically a data collection operation that then will take that data and sell it to somebody. So who are they selling it to? They're selling it to companies like Pfizer, and they're selling it to companies like Genentech. Those are the two biggest companies that 23andMe has sold their data to. I don't know if the amount that Pfizer paid is universally disclosed. Genentech paid about $60 million for genetic data that 23andMe has. So we're not talking about insignificant amounts of money here. But what are they doing with that data? So Pfizer paid this amount of money for the data. Genentech paid $60 million for their data. Are they just sitting on it? What are they doing with it? So they're both looking to try to find, again, do that bioinformatic research about finding particular mutations and then what diseases those could be related to. So in Pfizer's case, they were looking at Crohn's disease. In Genentech's case, they were looking at Parkinson's disease. So the other thing that Genentech did with this data is that they went back to the people that actually supplied the DNA, and they said, okay, so you supplied the DNA to 23andMe. We would like to do this study for Parkinson's. Can we have some skin cells, or can we have a biopsy that we can then take and also sequence? So in addition to getting the data, the company also has access to the people that gave the data, so they know who to go to ask them. So they've already been receptive to the idea of giving over some of their genetic material for something, so they might be receptive to other things as well. So it's really good for the companies, but you have to think about the fact that your genome may be the most personal thing that you have. Your genome is not necessarily completely unique to yourself, but it's pretty personal. And are you okay with giving over rights to somebody else, to a company? That's a personal decision. It may be good, it may not be good. I think it depends on who you're talking to.
Just full disclosure, 23andMe is not a sponsor of this show, but I actually had no idea. Do you think people that, again, I haven't gone through the process of using them, but do people that use them know that their information is I'm sure it's de-identified and all of that, but
Sure.
Well, maybe not. If they can come back to you, I don't know. But do you think people know as they're going through this that that's the case?
So I would say if you have an iPhone and every time the service agreement comes along, do you read that? Do you read those hundreds of pages of service agreement? I don't. Most people don't. So all of that information is there when you pay your $150 or whatever it is you pay now to 23andMe. And when you sign stuff, that's what you're signing. So I would say that most people don't know, but they don't know because they're not reading the stuff that they're being sent. I would say it's a lot like Facebook, where people are outraged. Oh, they have all of our data. Yes, but in a certain way, they've been telling you all along. It's just that it was either hard to find or people didn't want to find it, and so. Companies are relatively upfront from their perspective, and not necessarily from people's perspective, about what it is that they're doing. But it's up to people to sort of read and figure out what's going on.
Right.
Too.
No, it's interesting. I never thought of it.
Yeah.
Just going down that path for a second. So, aside from sources like 23andMe for pharmaceutical companies that are doing this kind of research, where do they get, if they don't use things like this, or where else do they get their data from? Is it just randomized controlled studies and things that they initiate, or where do you get large pools of data like that then to do that kind of research?
So you're talking about where do the companies get the research?
Yeah.
Or the data? So that's lots of places. Yes, the randomized clinical trials, human subjects that they will have in their trials for sure. One of the things that the Obama administration did was launch a large-scale precision medicine initiative before he left office, which is now, I believe, called All of Us because its name changed. But basically, their goal was to get, I believe it's 1 million people to get their genome sequenced, to have a large pool of data, public data for people to draw from. So. A couple of other places have basically done very similar initiatives. So there's several places to go get this information from, some public and some private.
Have you had yours done?
I have not, and nor will I ever get
Really?
mine. Yeah.
Why?
Partially,
what are you hiding?
Partially because of privacy issues. I am not incredibly comfortable with somebody having my genetic information. We do have laws, one of which is called GINA, which prohibits insurance companies from using your genetic information against you or whatever is found in there against you. But more than anything else, it's not statistically relevant. So going back to the example you gave a little bit earlier about your friend saying, I'm 1% this. That's one, in my opinion, one example of why you spend 70, 80, 90% of your time grooming the data before you do an analysis. So what is that actually saying, and what are these companies actually doing? So 23andMe, I can tell you specifically because they have this information on their website. They are not sequencing your genome. They're doing what's called a genotype. That basically means that they know specific spots of a genome where there are known mutations, depending on sort of who you are. Statistically significant mutations occur in certain groups of people, if that makes sense. So if you know that those occur in certain areas of the genome, you can test those particular areas. And so you're not sequencing those areas, but you're basically taking a very similar piece of DNA and hybridizing it and seeing if it matches up or not, and how well it matches up. So you're taking that data and then you're drawing some conclusions based off of it. So if a certain set of mutations are known to occur in a certain group of people at a certain statistically significant level, then you can say if you have this particular mutation with a certain degree of statistical relevance and realty, that you're probably this. And so you can see that I've used a lot of qualifying terms and all of that, which is not the same as saying you're 23% Ghanaian and you're 15% Scandinavian, which is a lot of certainty which really doesn't exist in the data.
I see.
So to sort of compound that problem, a lot of the information that is used to base these claims off of come from a sort of Scandinavian, Northern European, let's say Caucasian background. There's a lot of information for people that look a lot like me. If you don't look a lot like me, there's a lot less information that's known. So if you're talking about populations from Africa, if you're talking about populations from Southern Asia, or even Eastern Asia, there's a lot less information. So if you happen to be from a non-Caucasian background, the chances are that your information is not going to be as accurate as it would be if you come from Europe, or let's say probably most people in like America and North America.
Is that because that's just a limitation of the pool,
Yes.
the research pool?
Absolutely. And so there are a lot of efforts underway to try to collect a lot more data from people that are not Caucasian. Those have recently started, so they haven't had as much time to sort of take effect, so that data is a lot more limited. Yeah.
Wow, this is no idea. This is interesting stuff.
Yeah.
Back, I think you're talking about precision medicine. Could you talk a little bit about CRISPR? I know that I think that's related in the sense that from my limited knowledge, but
yeah.
talk to us about, explain what that is perhaps, and how it's used.
Sure.
And some of the issues around it.
So CRISPR is really an interesting thing. Where to start with this? So I also have somewhat of a personal connection to CRISPR, of all things. So thanks for bringing that up. CRISPR was initially discovered in prokaryotes, so in things like E. coli and microorganisms, and it was discovered in about 1987 by this guy with the last name of Ashino, and I actually know him, which is my personal connection. I actually know Ashino. He got all of his degrees in Japan, but then he did his postdoc in Dieter Sohl's lab at Yale, and that lab works on sort of the information and transfer apparatus within organisms, which is replication and transcription. So he discovered that E. coli had this particular set of clustered repeats that were actually able to sort of take care of viruses when they would attack the E. coli cell. So if a virus attacks a particular cell, this is basically the immune system of E. coli. And not only was it an immune system, it was an acquired immune system, and it could sort of learn a little bit too. So if it got attacked by one virus, it could then in the next time defend itself against that virus. And then if another virus attacked, if the same virus attacked, it was faster at destroying that particular viral DNA that would go in the cell. And then the same thing for new viruses. So for a long time, it was this acquired immune system in E. coli and prokaryotic cells, which was kind of neat because we think of ourselves as having an immune system. We don't generally think of small unicellular organisms as having an immune system, but they do. And it wasn't until 2010, when people started to take a look at this and say, oh, well, there's other things that we can do with it. So about 2012 to 2015, people had done a lot of work on this, and it was like the runner-up for like Invention of the Year for like several years, and I think finally in 2015 it was like Invention of the Year by Science Magazine. So this is now like 30 years or so after, 40 years after its initial discovery, that somebody had figured out, oh, we can do all this other stuff. And that other stuff primarily is modifying specific sections of the human genome. And so instead of targeting large areas of the genome to modify, you can actually get down to the individual nucleotide level and make small changes. And that's a pretty big deal. And you can do a lot of stuff with that, like take care of metabolic diseases and a lot of other things that are basically single nucleotide changes. You can change that with CRISPR and potentially, basically fix these people and make them, not have these metabolic diseases. But with everything, there's always consequences, and there's always these things that you hear about. Oh, all these potentials that you can use CRISPR for, you can do all this stuff for, but a lot less talked about. Well, what are the problems with it? So CRISPR's problems are that there are these things called off-target effects, which basically means that in addition to changing the one particular nucleotide you want to change, you also have these nucleotides that get changed over here as well, which were not expected. And then so what does that lead to? That could lead to a new disease that wasn't there already. So you may be fixing something, but you may be causing another problem at the same time.
I see. Previously you were talking about precision medicine. Is precision medicine related to CRISPR, or is it different? Is CRISPR sort of a tool that you can use to implement it, or is it a different thing?
So precision medicine really is sort of looking at individual mutations and trying to develop drugs, biologics, medical devices that target that specific mutation. And so CRISPR would be to look at that individual mutation and use this process to fix that just on the spot.
I see.
So you're not using a drug or a biologic that would then interact with something to take care of the disease. You're actually fixing the DNA sequence with CRISPR, so that doesn't happen anymore. So let's say you have a metabolic disease, and I can't think of one off the top of my head, but if you have, a lot of people have metabolic diseases, many of those are single nucleotide changes in the sequence of a gene. And so if you just change that from the wrong letter to the right letter, then you're done. You don't need to take any biologic, you don't need to take a drug, you don't need to do anything because you're cured. Assuming that everything works the way that it's supposed to.
Okay. Last new development I wanted to talk about,
Sure.
Stem cells, which have been around for a long time.
Right.
But I just wondered, I remember it was super controversial at the beginning.
Absolutely.
I hear people getting stem cell treatments all the time. What is it a concern? Is it has it evolved in a good way? What are some concerns about it?
Right. So I don't think scientists ever saw this as a problem. It was sort of a natural outgrowth of research and sort of a good way to try to do a lot of work. You'll probably remember in the George W. Bush administration, there was a lot of talk about limiting the use of stem cells, maybe possibly doing away with all embryonic stem cell research. So he basically vetoed a bill that would allow federal funding for embryonic stem cells, but then eventually that sort of got rolled back. He initially said that, okay, the only stem cells you can use are the ones that we currently know about, which seemed weird to scientists because basically every time you do an experiment, technically you're creating new stuff, and so that was sort of not a very workable solution. The Obama administration came along later, and I think later in the Bush administration as well, and rolled back a lot of that stuff from the early part of his administration. So there is federal funding for stem cell research. Most of it is with adult stem cells, but the other offshoot from the initial George W. Bush veto was that places like California and Texas said, okay, well, we're going to go ahead, and if federal money can't be given to doing embryonic stem cell research, we're going to go ahead and do it ourselves. So California and Texas both passed laws to allow embryonic stem cell research, and even Maryland actually passed a law to say that it was okay. And so there's a Maryland stem cell research initiative that's still going on to this day that was basically the product of this veto.
Wow. Okay. I didn't know. In terms of the effectiveness of stem cell treatments, are they do they work? Are people optimistic about the potential?
So I'm not going to claim to know about all of the things that stem cells are used for, but I will say that I'm not aware of any stem cell therapies. So I know that I have friends, let's say, that have knee problems, and their doctors are like, oh, well, part of the things that we'll do is basically inject you with stem cells, and maybe it'll help whatever knee problems you have. So you have things like that. I don't know of any of those type of approaches that are FDA approved, and the FDA has a big role to play in all of this. We very much need the FDA for this type of stuff. So I'm not aware that any of that is legitimate. But stem cells
people aren't sure that it works, they just are trying it.
Right, exactly.
It's like in my big fat Greek wedding, they have the Windex. Just spray everything with Windex.
That solves everything.
Hope that it does it. That's how it is.
Exactly. And I can't say that there aren't people out there that it works for. I'm more than positive that there are. Will it work for a large proportion of the population? Probably not. What approach do you take to find out if this works? Do you have a doctor or somebody just inject you with something? Does it work? Does it doesn't work? Or do you go through the sort of scientific, rigorous clinical trial approach where you can say for this particular population it works this amount of time, and for this population it doesn't, and do statistics, significance and categorize all of that? I think that latter approach is the best, which is why FDA is so necessary.
Wow, interesting. So I want to spend a little bit of time shifting gears and talking about maybe some of the cultural or ethical issues related to your work. It's just such a fascinating. It's just such a fascinating area, and one that people wrestle with a lot.
Yeah.
And one of the things that's always been, on my mind is looking at a distinction or maybe blurry line between the use of biotechnology to promote health versus enhancement. And I think, we all, of course, when it's your own health or the health of people you love, people, it would seem, would do anything. But but there is a kind of a line with the things that to make you better. Steve Austin, make them better. But when it's you or the people that you love, maybe it seems less concerning. But could you talk a little bit about that? How is this addressed in the field? Is there a literature on this? How are people thinking about these issues?
Right. So A little bit of it depends on what you mean by enhancement. If we're going to go back to the beginning where you talked about the $6 million man and you talked about, cybernetics and, prostheses and adding limbs where people don't have any, that can be considered enhancement. I think that that's pretty well accepted, that's perfectly okay. I don't think anybody has any general problems with that. If you're going to talk about biohackers and bioengineers, who are these people that basically want to try everything and see sort of how that works. Maybe a little bit to some extent if you're talking about Michael Pollan's new book about looking at these sort of LSD type compounds and sort of what they can do for people, whether they can heal people or not. There's a little bit of research on that. But more than anything else, I think that what this highlights is the need for a place like the Food and Drug Administration and why it's so important in everybody's daily life. We forget these things because most of us that are alive now don't remember a time without the FDA. But there was a time, 100 or so years ago, maybe a little bit more now, when you could sell anything, you could tell anybody anything. I mean, think about the derivation of the word snake oil. That's where all of this stuff comes from. You could basically just sell whatever you want to. You can give people whatever you want to and say whatever it is and say it does whatever you want it to say. The federal government decided after a while that it was important that whatever we were selling to people, first of all, be safe. So that was the first level. It didn't have to work, didn't have to do anything, but if you took it, you weren't going to die or you weren't going to have some sort of horrible side effect. And then after that sort of level was established, then people decided, oh, well, in addition to being safe and not killing me, this thing should actually do what it claims to do, so it should be efficacious. And so that was the second level of regulation that was developed, which led to the modern part of the drug part of the Food and Drug Administration. So this is an interesting part of all of this. Should you allow people to do whatever they want to themselves? They're only hurting themselves, probably. Is that part of our basic freedoms as Americans, as human beings, that we should be able to do whatever we want to ourselves? I can't say that that's necessarily wrong. I will say, though, that if you want anything that you're doing to benefit somebody else, the best way to do that is in a thoughtful, scientific, rigorous way where things are cataloged and it's tried on somebody else other than yourself in a thoughtful, cataloged, statistically significant way, so you can make sure that whatever it is that's happening benefits more people than just you. So from a certain perspective, biohacking is a little bit egotistical because really, honestly, you're only concerned about what it does to you and not other people. Those of us that are in biotechnology who want to see drugs and biologics sort of passed out and given to as many people as possible to help them, it's more of a sort of, an ethical way of doing it and helps more people.
It's your program you're program chair of a program on biotechnology,
right.
Regulatory affairs.
Correct, yep.
So that must deal with this whole category of issues about human control, government control, boundaries of these things.
Right. So one of the well-accepted things in the biotech community, the pharmaceutical community, is the fact that FDA needs to exist. It's one of the few spaces, the few sort of business spaces where government regulation is well accepted and wanted. And one of the interesting things I will say, it's getting off topic just a little bit, is that one of the things that's coming down the pike that's actually really already here a little bit is the interface between technology and medicine. And so you have this clash that's starting between places like Silicon Valley and the biotech community. The Silicon Valley people are not used to being regulated generally at all, or very little, if at all, regulated. The biotech community and the pharmaceutical community, on the other hand, are heavily regulated, and not just heavily regulated, but it's heavily regulated because they want to be, because they see the need for it. But the Silicon Valley people think that that's completely unnecessary, and they're running into this clash of, we want to get into the health sector, but we're not used to being regulated, and so they're bristling a lot at being regulated, and they don't really see the need for it, but there's a very large need for it. So the regulatory...
That's a very timely debate, though. I think one of the things I wanted to ask you about was about how we view, comparing kind of healthcare technology with other kinds of technology. Certainly, for example, we love our gadgets, but I would say in the last year or two, because of things that we've seen, concerns about privacy and hacking and even other governments participating in this, I think that there's been some skepticism and some caution about it, and the perception that regulation is a nuisance and a hassle might change even with some of those things. But so you're saying that in the healthcare area, there is this natural caution about these things, whereas I think with other kinds, I think with social media, I think we've kind of lost our innocence a little bit with that. But you're saying that in healthcare there has been a caution about it from the beginning?
Right, so it's interesting that you sort of bring this up. So to connect that to the Silicon Valley talk that I from earlier, if you think about Fitbit, everybody's got, I shouldn't say everybody, most everybody has a Fitbit, right? So what is that measuring? So if somebody looks at their Fitbit and they say, I did 50,000 steps today, my skepticism says, did you? And how do you know that? And the usual answer is, well, my Fitbit tells me 50,000 steps. Okay, but what is that measuring? Well, it's measuring my steps. No, it's not measuring your steps. So this is what I mean.
You're a
by
real party pooper, aren't you?
I totally am a party pooper. So I will also mention that I know a lot of people that work at FDA. Fitbits are not regulated or cleared by FDA, but everybody that works at FDA has got a Fitbit. So this is the wonderful thing about life. So what is it measuring? It's measuring basically either this motion or your legs going back and doing the same thing. Are those steps? No, because I could sit here and I could do this. And if you want to drink, you could drink 50,000 steps depending on how much you want to drink during the day. But is it helping you to get up and to be more active? Probably, because that's generally what they're for. So it's doing what it's designed to do, but it's not giving you truthful information. And that's where that clash comes in between Silicon Valley and health. So for health, if you're making a claim, it's got to be true. But Silicon Valley is not used to that. They're used to making gadgets that save the world. I don't know that gadgets or apps are really actually literally saving the world, which would be the truth part of that, but this is the clash. And people in health and people in biotech take these things incredibly seriously because, again, we have that history of people dying. You think about like the thalidomide babies in the UK. The reason why you don't have thalidomide babies in the United States is because of the FDA. And the FDA was established before thalidomide was used widely in pregnant mothers. And that got stopped in the United States. It got approved in the UK, but it got stopped in the United States because of the FDA. So health-related issues, privacy issues, these are things that are treated very seriously by the field, for sure.
I had no idea, actually. That's really, that's amazing. And the intersection of these two forces is something I'll pay much more attention to. We're just going to wrap up in a minute, but I wanted to ask really about some of the ethical implications. I mean, so much of the science and the thought about the tools and the technologies have done amazing things to improve our lives. And the lives of people in general. But is there a branch, but there's also room for excess as and boundaries as you've pointed out. Is there a part of this field that deals with what it means to be human and the humanity, the dignity that humans carry?
Sure.
Or is that just something that people consider that's a sort of religious question or a personal question? Is there a part of the field, and are there groups and commissions and organizations that think about that in a serious way? Yeah,
yes. The short answer is yes. The longer answer is, in my beginning, I didn't mention the fact that I have a minor in philosophy, so now we're getting into my other thing that I like to talk about. So the field is called bioethics. There are a lot of bioethicists that are out there. There are bioethicists at NIH. There are bioethicists at probably most research universities. And that's one of the many things that they spend their time thinking about and talking about is sort of what does it mean to be human? Is it okay to do this particular type of research? If we're going to talk about the stem cell research earlier, that's part of the debate. Is it okay to do these things? If we're going to talk about CRISPR again, is it okay to modify the germline of human beings? So, humans have two basic types of cells. They have cells that are in your body, but then also the cells that make up sperm and eggs that go on to make another human being. If you make a modification in one of those or in an embryo, that gets passed on, and you'll continue to pass that on. Is it okay to make a modification that gets passed through? Because at that point, you're talking about the Aldous Huxley designer baby. Is that an okay thing to do? So a lot of these things get debated about whether it's okay, whether it's not okay. I don't think that you can stop science. Even if you stop the funding, there's going to be somebody somewhere that wants to go ahead and do this. But you can sort of control it a little bit through regulation, through talking about it and trying to be as open as possible, and having people like the bioethicists sort of think about the other side of things. Since you brought up religion, I will also say that as a scientist, I don't think that science and religion are mutually exclusive of one another. I think that they answer different questions. That science answers how questions and that religion answers why questions. So I can tell you as a scientist how something happens. And that's generally what I'm trying to do, is trying to figure out how does this happen. Why that happens, that's not a question that I can answer. That's a question for somebody else to answer. The why part is totally separate from how.
Well, I think that's a good place to end it. We started off at the low end with $6 million man, and we're ending up with religion. So I think we've gone from the low to the high. So thank you.
Absolutely. It's great to be here.
Professor Coker, and we hope you'll come back and talk more about this.
Absolutely.
Appreciate
Love to.
it.
Yeah,
Thank
thanks.
you.
Yep.
Thanks for watching Thursday Thoughts with Dr. Alan Drimmer. This has been a production of University of Maryland University College.